Okay, well, we're 3 minutes past 10. I'm going to get started, and I expect more will continue joining over the next couple of minutes as well. So firstly, thank you very much, everyone, for registering and attending the third FHERITALE webinar, today focusing on untargeted approaches to marine pollutant monitoring. What I'm going to do first, before handing over to our speakers, is just give a brief overview of the aims of the project, and also some notable outputs that might be interesting to some of you as well. So, the core aims and objectives of FHERITALE. Well, firstly, FHERITALE stands for Food, Health and Environmental Research Infrastructure to tackle emerging priorities. And what you can get from that is the project aims to enable the research community and empower the research community, in fact, to study and analyse the impact of artificial materials, including things like micro and nanoplastics, on life using a One Health approach. So this means both taking into account direct and indirect effects on things like health, food and the environment. And the project aims to address this via a number of key goals. The first of which is mapping the services available through European research infrastructures to researchers seeking to conduct research in this area. And this is something that the project did relatively early on. And as part of this, partners also identified gaps in service provision and key technological needs to researchers within the community as well. Of course, following that, the project then will aim to make strategies for increasing service capacity, both in the area of services that already exist, but also in addressing the gaps that have been identified both in service provision and in key technologies as well. And perhaps most importantly, promoting an interdisciplinary approach to research within this area, within this One Health context. So ensuring that researchers can access services across research infrastructures, and we'll be making plans to promote this in the near future as well. So collectively, we hope this will provide the scientific community across Europe with easy accessible services at a single entry point via this one-stop shop. But it's important to note that FHERITALE won't fund this research. It will help researchers access pre-existing services, help research infrastructure serve new communities, and also provide input via things like policy briefs for future funding priorities based on the needs of our communities. So this is an overview of the partners that are involved in the project, and you can see they're spread across Europe through 4 different research infrastructures, namely Instruct, based in UK, but also in Italy and Spain. And the project is coordinated by Chirm, based in Italy, in Florence. Also Metrofood across 3 distinct sites in Europe, Untargeted in Paris and France, and also Irene across the Netherlands and Czechia as well. And you can see also the project is due to finish at the end of this year, so we hope to have some exciting outputs coming to you over the next 9 or 10 months as well. So just as some key outputs today, early on in the project, it was necessary to define what the key priorities of the project would be, because obviously artificial materials is a relatively large area to tackle. And based on a very detailed and in-depth bibliometric study by some of the partners, looking at the landscape over the previous 5 years, 4 key thematic priorities were identified, namely micro nanoplastics, bioplastics, plastic additives, and metals and metal oxides. And then based on these, we conducted a detailed analysis and generated this landscape and directory of services that are available across European research infrastructures that address these core focuses. And you can see from this quite busy schematic that there are a number of research infrastructures offering services across the services on the left-hand column. And if you want to see this in more detail, and indeed a slightly bigger version, you can go to the FHERITALE website and look under our outputs where you'll be able to find this. And perhaps more recently, we've also been conducting things like gap analyses and identifying the priority needs of European researchers working within these fields. So we've conducted surveys and in-person discussions relating to key service and technology needs within the community. And you can see a breakdown of some of these priority needs on the left-hand side here across food, health and environment and cross-domain applications. And you can also see thematic clusters that have been defined on the right-hand side again, across a number of different areas. And if you want to look at this deliverable and this report in more detail, you can follow the QR code. And I should say this webinar is going to be recorded, or is being recorded, so you'll be able to see these QR codes on the recording as well. And I'll share the slides of this presentation. So other ways to stay updated, of course, you can go to our website where you can see key outputs and relevant news and information, but you can also follow us on both LinkedIn and Blue Sky, where we post relatively often and stay up to date. There'll also be an opportunity to join us in person in Brussels on May 26th, where we're going to planning to host a stakeholder meeting focusing on cross-sector synergies amongst research infrastructures to strengthen European research on the impact of micro and nanoplastics specifically. And there's more information by the QR code there. And with that, I will stop for the moment, and I will stop sharing my screen. And I will hand over to Dr. Didier Stien from Sorbonne University, who is going to talk to us today about some of his work over recent years. And Didier, I'll just give you a brief introduction and then you'll be good to go. So Didier joined CNRS in 1999, initially working on natural products inspired by observations on species networks. But since 2013 at Sorbonne University, he's also focused on bacterial and fungal symbionts in marine organisms such as plants, algae, and sponges, again, with the aim of discovering innovative, active natural products. But more recently, he's focused on toxicity and fate of emerging pollutants, including the search for toxicity markers in marine organisms and the biotransformation products of the pollutants tested. And it's this work that Didier is going to talk to us about today, and we're very excited to hear more about it. So with that, I will stop talking and pass over to Didier.
Okay, thank you, David.
We can see the screen, that's great.
And you can hear me?
Yes, we can.
Okay, so I think it's all right.
We're good to go.
So thank you for the invitation to talk today. It's first of all, I'd like to say that I like very much the acronym FHERITALE. Congratulations for finding that. And I'm here to talk about our work on how we try to unravel the fate and cumulative impact of marine pollutants. And I'm a CNRS researcher in a lab that is associated to to Sorbonne University, University of Perpignan, and CNRS in France. So just a short comment on the exposome concept. This concept was introduced in 2005 by the British cancer epidemiologist Christopher Wilde. And the idea is to better understand the influence on health of all the exposures that an individual experiences through his entire life, from the conception to death. And the exposome includes chemical agents, of course, that you are exposed to with your environment or your food, physical agents, noise or wind, and biological agent, microorganism, for example, nutritional deficiencies or socioeconomic factors such as stress. And basically, what it tells you is that if you have predisposition, a genetic predisposition for disease, but you don't have anything else, then you may not develop the disease. And if you have cumulative exposome but not genetic predisposition, you may or may not develop a disease. And with the two together, this is how the disease might come. And that's for an individual. But this reminds us really what we see in the environment with what's called the cocktail effect, or the eco-exposome. And in our group, we are interested in the pollutants, mainly the organic pollutants that come into the coastal waters. And those have an originate from human activity on land, of course, but also from direct activity into coastal waters. As I said, we are mostly focused on organic pollutants, and since we've worked with and discovered that some marine organisms can transform some organic pollutants into compounds that are going to accumulate in the food chain, in their body and in the food chain, we're also interested in the fate and the transformation product of these organic pollutants. So what we need is to characterize the eco-exposome and to quantify the pollutants that are involved in this mixture of pollutants from legacy contaminants or contaminants of emerging concern. But also, since some compounds are transformed or are just not expected in this environment, we want also to address the question of unknown compounds or the ones that would not be normally expected in the coastal seawaters or in the marine organisms. So we need to collect samples, and for that we focus, well, we want to have samples of pretty much everything, including marine organisms. I'm not going to talk about that right now, but so we get seawater, we get sediments, and we have marine organisms, and we want to look at the pollutants all together. And this is through metabolomic-like approaches and targeted analysis. So the project I'm talking about now is the CClean project, and it involves two laboratories, ours in south of France and another one here in South Portugal, colleagues from CCMAR. And these two locations where we collect samples are highly impacted by summer tourism. There are small cities, but there are a lot of people in summer. There is a little bit of urban runoff also. There's a lot of agriculture and viticulture. There's a very famous wine in Banyuls, and that has an impact on the environment too. There's riverine transport, more in Portugal. And of course, navigation. And what's interesting in these two places is that there are protected areas around, and they are said to be pristine, but I don't think there's any pristine environment on Earth now. So sampling sediment and marine organisms is easy. Sampling seawater is another question. We decided to use passive sampler for that. First of all, because they are recommended by the European Water Framework Directive. And we use different phase. Basically, they are like tea bags. You have an absorption phase in between two membranes. And the phase can be HLB. HLB is interesting because it absorbs. It's supposed to be specific to pharmaceutical and pesticide, but basically it takes anything that is not polar enough to remain into the water. And we also use a PFAS phase. Since it's specific, then it's not really untargeted, it's semi-targeted analysis, and glyphosate phase is interesting because of glyphosate, but of course this is really targeted analysis on glyphosate. So I'm going to focus here on HLB, the work that we have on HLB phase. And passive sampler are interesting for several reasons. First of all, I mean, the contamination may not be the same over time, and you put passive sampler in seawater for like 2 weeks, and anything that was present at some point in these 2 weeks is going to be captured by the passive sampler. What you get in the end is not a maximum concentration at some point. This is what you have with grab sampling, but if you take grab samples and the peak of pollution is not there, then you don't have anything. But what you have is the time-weighted average concentration. So it's not when I will give you concentrations later, it's not a peak concentration, it's an average concentration over time. And this is really important because if you see toxicity based on average concentration, then you can be worried for the environment. And the only drawback of this method is that you have to get sampling rate for, sorry, you have to get sampling rate for any single contaminant that you are going to find. But in the end, you get enhanced monitoring of diluted and marine environment because the passive sampler is going to concentrate the pollutants. So what we do once we collect the passive sampler is just a chemical profile analysis. This is done on the Biodemar platform in Banyuls. We have very cool equipment with SC coupled to high resolution MS. And we get that sort of profile here with a lot of peaks. Basically, one peak, even the very small ones, is one contaminant. And you would imagine that nowadays there are tools that just give you a list of molecules from this chemical profile, but this is not the case. We use some automatic analysis, but it doesn't work very well, and there is a lot of brain juice that we have to put in this analysis, and we do a lot of manual annotation of the different chemicals. This is something that we do well in our group, but we are trying to use this experience that we have to develop different things, and I'll show you later. Once you have characterized your compound, you need to quantify it, and this is done with, as I said, measurement of the sampling rate in laboratory. Then you generate data that enables to assess the environmental risk for the contaminants that you found in your seawater samples. So this is what we do with automated analysis. We have, if you take Banyuls and Polil Bays, for example, they are 5 kilometers away, one from the other. You have nearly 2,000 features. Features is an ion in the analysis, and most of them are common to the two bays. You would then you can start looking at what's in these features. Only the one that have MS spectra. Those are the only ones for which we can go further because we have more structural information here. And this is what gives you with an automated analysis tool that's called Canopus. It's giving the class of the compounds that we have. And first of all, you can see that 1/3 of them are just classified as unknown compounds. And the other ones, we have compounds that are natural ones, like amino acids probably, and we have clearly some pollutants like polyethylene glycol. But first of all, 1/3 are not annotated at all, and for the rest, I can tell you that it's actually not very good. And secondary alcohol, for example, is very vague. So after that, we really have to go into detail and annotate manually what are the major components of the mixture. So if you do that, it takes time. I'm going to show you what we have on 50 compounds here. We have 37 surfactants. 2 of them are quaternary ammonium. These compounds are present in laundry, detergent, and cosmetics. And I mean, there's a large concentration of these chemicals, in particular TTAB, and TTAB is from laundry detergent. And the reason why we have that, and we did not expect that initially, is that many people in Banyuls have their washing machine connected to the rainwater system. This is not allowed. This is out of the law, of course, but yet it still exists, and this is bringing a lot of these contaminants in seawater. These two are interesting too. Nonoxynol and peg-trisiloxane are agriculture surfactants, and nonoxynol is one of these surfactants that's used for the formulation of glyphosate. So we don't have yet the results with glyphosate MIPS that we've been using for in our passive samples, but we already know that there might be some glyphosate in seawater. We have 3 natural products among the major compounds that we have in our analysis. One is fucoxanthin, the other one lysophosphatidylcholine, there's another one as well. And these 2 compounds, the 3 actually, are from microalgae, so it's not so surprising to have this natural product. And we have one pesticide, DEET. It's used as a repulsive agent against insect bites. We have one dye, gentian violet or crystal violet. If anyone knows why we have this in seawater, I'm interested because I have no idea where it might come from. It's not allowed, it's not sold in Europe, I think, and it's not allowed in any application. So I don't understand why it's there, and you will see that its concentration is troublesome. And we have 9 UV filters that we were expected to find in coastal seawater in Banyuls. We have dipheniguanidine. That's funny. It's a rubber and polymer additive, and it's actually probably originating from car tires. And we have one compound that is it's funny as well. Of course we did not expect to have something like that here. It's this carboxylic acid, and it results from the hydrolysis of this UV filter that's called octocrylene. It's known from the literature in Burry's work that octocrylene is, when you put it on your skin, it's going to go through the skin into the bloodstream and eventually hydrolyzed in the liver, and it's going to be excreted in urine as this compound. So it means 2 things. First of all, it's an indirect demonstration of Burry's work. And second, it also means that people pee in seawater. I mean, I have a lot of things to say about octocrylene, but just quickly, you may not want to have octocrylene in your cosmetic products. If I already banned octocrylene in my house, and it's toxic for the environment, and it's probably also toxic for you when you use it. So it's still present in a few cosmetics in Europe. It's been reduced a lot. And I don't recommend that people keep using octocrylene. So we had 2,000 features, if you remember. We have 50 compounds for which we have a confirmed annotation, a structure basically. And out of these 50 compounds, we were able to get the concentration of 10 of them. It's complicated to have the sampling rate. And the concentration in seawater ranges from 10 nanogram per liter for gentian violet to 14 microgram per liter for this UV filter here, BMT. So the concentrations can be very high. And the question is whether these compounds at this concentration pose a risk to the marine environment. And the answer is we don't know for those 3 that are at the highest concentration. They are UV filter, but there is a really insufficient ecotoxicological data on this compound. So it's really urgent to see if at this concentration, this compound might have an impact on marine life. If you look at the others, octocrylene at this concentration is toxic for the marine environment. Gentian violet is toxic as well. And the mixture of the 2 quaternary ammonium that I presented before, the one from the washing machines, are toxic to the marine environment. So it's really a problem to have these compounds individually at this concentration in seawater. If you imagine that 3 out of 10 compounds are toxic to the environment, and you think that we had initially maybe, well, 2,000 features, that's less compounds, but it's not that, it's a lot more than 10. You can imagine that maybe the seawater is the animals that try to live in sea, or the plants that try to live in seawater in Banyuls, they are not, they are suffering serious difficulties here, and that really questions the cumulative impact of this mixture. What can it be? So this is how we are, and we are able to annotate manually a lot of contaminants that are really present in the environment that we are sampling, and this manual annotation are not lost to science because we are creating a database of contaminants that may help us later to annotate a lot faster all the contaminants that we find. We implement this database with commercially available non-pollutants like PFAS, drug, and pesticides. And the idea with that is to build a molecular network that will point out very quickly the non-compounds that we have in an environmental sample, and will also contribute to the discovery and the annotation of unknown or unexpected compound. And for that, we are building a web-based tool that will be called Annotics. It's not available yet, but hopefully, let's say if you invite me again in 2 years, I can talk to you about Annotics. And it just remains to thank the people that are contributing to the CClean project. So there is a CCMAR team in South Portugal led by Deborah Power and these people in Banyuls. Sorry, I don't have a picture of Nicola, and this guy, I don't have a picture as well. I need to thank EMBRC for supporting this project research, and you for your kind attention.
Thank you very much, Didier. It's very interesting. Like I say, if there's as John has mentioned in the chat, if anyone has any questions, please do put them in the chat or the Q&A, and that perhaps I can ask a couple of questions at this point. So you mentioned it sounds like you're essentially developing a database of reference spectra for these pollutants, and I wondered, is there anything that's existing currently, or because presumably there's a very large diversity of these compounds, and so how does your work fit alongside something that already exists, or is this something that's new?
Okay, so the database, there are databases of MS2 spectra. What we need is an MS2 spectra, right? There are databases out there, but first of all, they are not. We can use them, and we currently use them for the annotation, but we would like to have one that is the spectra that we have on our system, because the annotations will be more accurate with that. But more than just a database, we are building a tool that is going to use this database and the commercial ones as well to help in the annotation and the quick discovery on even unknown compounds. Yes. So this is our goal, but it's not ready yet. It's a lot of data that we need to dig in to get all the information. Absolutely. But you can see that with just 50 compounds, we already have a scary result, I would say.
No, completely. And so the idea would be with this Annotics tool that users can submit their results, and it will annotate the compounds in their findings.
That's the idea. Yeah.
That's very interesting. Yeah. Okay, and then another question I guess these techniques are in in the context that you've found in your research, have detected pollutants from surfactants to pesticides, etc. I wondered from obviously from FHERITALE, one of our priorities is plastics, microplastics, and presumably plasticizers and things like that are leaching from these substances. Is that something that you would be interested in including in the database, or is it something that you find often in your samples?
Yes, of course. We are interested to include that in the database and in our work, in our research. Some UV filters are also used in plastics. So right. It's partly already there. Yeah. And we have also, but those are more contaminations. For example, a colleague sent me coral eggs that I extracted, but they were kept in plastic vials. And I know exactly what are I had some plastic leaching in the coral eggs, and was able to annotate those. So some of these molecules that you're mentioning are already in our database and were annotated manually by us.
Okay, no, that's great.
But we can, of course, we need to. The more we have, things in the database, the better we are.
Sure, absolutely.
Actually, the analytics tool does work already. It's just not yet available to the public because it doesn't work with commercial database, but it does work with our own database.
No, that's great. That's really good. I can see a question in the Q&A from Ryan. So for the pollutants you've identified in the bays, on what timescale would you expect them to break down or be removed from the environment? Or would they persist?
Sorry, I'm trying to find the
in the
place where section. Well, that depends really. To say the truth, it depends. For example, crystal violet or gentian violet, whatever. I don't know why it's there. Maybe it's old use, or maybe it's used in other countries, or maybe it's illegal use somewhere. But I mean, we have it at the exact same concentration at all period of time, and in the two bays that we have analyzed, and they are 5 kilometers away. So I don't think it comes from Banyu's city, because it's not it's present the same in Pauline's Bay. I don't know if how spread this crystal violet is in the Mediterranean seawater, but if it's the same concentration everywhere, then it's a bit scary to see that. And if it comes from far away, then it's probably a low concentration that we have. So, and that would mean that it's really stable in the environment. If it's an old use, like some compounds can be, like the DDT has been banned for decades, and we can still find it sometimes. We don't find it in our samples, but in other environmental samples, it's possible to find DDT. Some compounds are really like the PFAS would stay like forever. They would probably accumulate in different compartments. PFAS are really not soluble in water, so you would probably see them in the sediment or in marine organisms. There are very few bacteria that can degrade PFAS, so it goes very slowly. And the question is, the answer to this question really is depends on the nature of the contaminant.
Yeah. No, that makes sense, given the diversity. Brilliant. Well, thank you very much, Didier. If anyone has any other questions or wants to follow up on those, do feel free to type them in the Q&A. And if we have time at the end, we can come back to them, or maybe Didier can answer them in the chat himself if he's able to. But with that, then I'd like to move to our second speaker, Professor Carlos Cordeiro. So Carlos, I'll just give you a brief introduction, and then you'll be good to go. So Carlos is currently Associate Professor at Universidade de Lisboa, where he coordinates the Portuguese mass spectrometry network and also co-coordinates the Portuguese research site of Instruct-ERIC. The main topic of research Carlos focuses on is the development of extreme resolution mass spectrometry, or magnetic resonance mass spectrometry, also known as FHERITALE, and its application to metabolic fingerprinting from microbial cells to humans, human diseases, and also in structural biology. But Carlos will be focusing away from structural biology for today's talk. So I'll hand over to you, and we look forward to hearing.
Thank you, John. Thank you, David, for the very nice introduction. I'm sorry.
No problem.
I've been used to deal with John for quite a few years, and in Instruct, and you are new to me at least. So I'm now sharing the presentation. Can you see it?
Yes. Yes.
Oh, perfectly. So indeed, I do work with a very specialized type of mass spectrometer that was formerly known as Fourier transform ion cyclotron resonance mass spectrometer. So currently it's known as magnetic resonance mass spectrometer. So basically this looks like a NMR that has been tilted over, and physically it's actually very similar to an NMR because it works on the time domain to the frequency domain. So it's basically a trap, a magnetic trap. And the ion dynamics is determined by the Lorentz force. So basically, these are two installations. This is the one in Lisbon on your left, and you have the other one on the right, which was installed last year in Rouen. And the main difference is actually the magnet. So you have a higher magnetic field in Rouen, 18 Tesla versus 7. So these have some implications in terms of resolving power, dynamic range, but the effect is not as pronounced as in NMR. So basically, qualitatively, the results are always very similar. So what this instruments allows us to do is actually to have a very, very extreme resolution. So this allows us to deal with very complex mixtures without actually using separative techniques. For example, on the left, you have a sample from crude oil, and crude oil can contain up to 400,000 different components. So this is not feature level, this is identifiable formulas. And just zooming in, you can see there's a lot of detail in the spectrum. So this would be the most complex type of samples that are dealt with. On the other hand, if you're dealing, say, with samples that are extracted for something that was alive, at least before the extraction, so on a metabolomic sample, what you actually have is a different picture. So you have more sparse regions of very high density with regions of low density and a very pronounced effect of the dynamic range. So analytically, the situation is not exactly identical, but it's still manageable. So how do we transform this kind of complex spectra into chemical, biochemical, and biological information? So for that, I will show you an example. This is actually an analysis from the traces that are left back when a finger mark is deposited. For example, if I touch the camera, you will see my finger mark. This finger mark is actually a very complex mixture made out of exposome components, endogenous substances, things that I may have or the person may have had for food, or imagine that person may have manipulated explosives, taken some drugs, some medicine, and a lot of these things will show up in the composition of the finger mark. So if I enhance a region of the very complex spectra we have, we can clearly see the isotopic distribution of carbon. This you can see on any regular high-resolution mass spectrometer. But what you can only see on a magnetic resonance mass spectrometer across the entire range is actually the so-called fine isotopic structure. So if you look at the carbon-13, you will see there are tiny little peaks here that actually match deuterium and nitrogen This shoulder is oxygen. But if you go to the third peak, you'll see a few more things, and the fourth peak, and so on. So if you have this, and if you have, say, a mass accuracy that is sub-ppm, you can actually obtain molecular formulas very accurately and totally independent from databases. So this kind of ab initio calculation is what magnetic resonance mass spectrometry provides. And from this on, you'll use this information to, through curated databases, get information about what are your compounds. So we are completely skipping fragmentation and any kind of analysis. And the main reason is because at this scale, we are dealing with tens of thousands, lots of tens of thousands of signals, and it would not be feasible to actually do MSMS of all of this in a realistic time frame by direct infusion. So basically, an analysis for one of these spectra can take between 5 to 15 minutes, depends on the resolution we want to achieve. So it can be very, very high throughput. But one thing we notice is when we go to curated databases, even if chemistry tells me that a lot of isomers are possible, not everything that is possible actually happens. And we start noticing that, for example, if you look at the situation where we have 32,000 signals that we can annotate, say, 1,500, we can actually see that about 40% of the annotations correspond to just one possible structure. Of course, we have very poor situations where we have things that can have 825 possible structures. But in between, the vast majority has less than 5 annotations. So we start thinking about what can we do with this information, and we actually came up with an algorithm that actually ranks, scores, and ranks our formulas according to the likelihood of that formula being a certain structure or a certain compound that we can annotate. So this involves not just accuracy, fitting the isotopic pattern, but also correlations and other techniques that we also use to do this rank. So basically, we use what we call single-shot metabolomics. From a tiny amount of sample, zero preparation, we dilute, infuse, and then, of course, extract information using computational tools. Of course, we can use commercial computational tools or freely available on the web, but for best results, definitely we have developed a software called Nemesis that actually implements the entire workflow. So basically, the workflow goes from the transient, that means the raw signal that comes from the mass spectrometer that needs to be Fourier transformed, apodized, and so on, to the full analysis, including several types of network analysis. For example, mass difference and formula difference networks. So these are important for us to realize what kind of biochemical or chemical processes are actually happening in our samples. This is a very powerful technique as a classifier. For example, what you have here is metabolic classification of different yeast species. And this would be a typical phylogenetic tree based on genetic information. And of course, if you look just at the metabolism, you can see that they're clearly separated. Of course, they are separated in a different way that you get from genomic information, but all in all, this is a very accurate technique for identification. It also allows us to work in a field that we called metametabolomics. That is, can you identify what was the living being that was present in a certain situation just from the chemical traces that were left behind? And it turns out it's possible to do that. So these are 3 soil bacteria, 3 different species from Pseudomonas, and 1 species of Bacillus. And you can clearly see, just on a very simple statistical analysis, that the clustering clearly separates these 4 species. And if you look at this dendrogram, you'll see that the first bifurcation immediately separates Bacillus from Pseudomonas. So this has a lot of potential for identification and for fingerprinting. Now that we know how magnetic resonance mass spectrometry works and what we can do with it, I will now guide you through a journey to one of the most pristine areas on the planet. Actually, I don't go there. I work with biologists and other people that go to Antarctica. And this is a view of Deception Island. I would say a most fortunate or unfortunate name to choose for an island. So this is a very remote area of the planet. So we should expect to find clear waters, absence of pollution, not like you found in the south of Portugal or the south of France that are completely. I don't think next time I will go to Algarve, I'll think twice before swimming in the ocean again. Or definitely, I will try not to sip any water while I swim there. But Antarctica, unfortunately, if you see at this picture, you can see there are debris, there are people walking on an ice beach, and in the horizon, you can actually see a cruise ship. So we started by collecting plankton, phytoplankton, and looking at the metabolism. Actually, the study was not meant to find pollutants, but when we were annotating and investigating what we found there, we found something like 70 compounds that should not be there. And all these are persistent organic pollutants. So these things, I cannot say nothing is forever, but these things are likely to stay there for quite a long time. And if you look at the classification, you can see there are a lot of biocides, but you also have this polycyclic aromatic hydrocarbons that come mainly from fuel. And you also see a lot of pentachlorophenol. And this is worrying because this is an herbicide that has been used from the 30s. So definitely from the 30s to now, from last century to the 30s of this century, so in about 100 years, we have this herbicide all over the planet, which is quite worrying. And the vast majority of things are actually things of human origin, things like you recognize, like ibuprofen, phenazopyridine, and for example, enbolin. Enbolin is found in all samples that we tested in some cases, so this is the frequency with which we detected. And in some cases, for example, 1/3 of our samples contained thalidomide, which definitely it's a highly mutagenic substance and is not something, first, it's not something that you want to take in tablets, and it's not something that you want to find inside phytoplankton, because if it's in phytoplankton, it's in the food chain everywhere. There will be a dilution effect, of course, but it's there definitely. So then we went on the next step. And what was our next step? Our next step is what kind of effects do these compounds actually have on this kind of organisms? And for that, we use two approaches. I'm just going to talk about one of them. We use, this can be done, I mean, you can have fishes in the aquarium and just throw in the substance you want to analyze. You can do it with fish cells in culture, which I think is probably the best model. So you can actually use hepatocytes from fish and test this, or you can actually do this with phytoplankton. And this is exactly what we did here with the fluoxetine, which is also known as Prozac. And this thing has been detected in seawaters at concentrations that in some cases can reach as high as 40 micrograms per liter. Usually the detectable concentration is much lower, is usually in the nanogram to picogram, but what is quite worrying is that I think this kind of substance is detected more or less everywhere. And I'm pretty sure that in the list of annotated compounds that Didier just showed, I'm pretty sure this thing is there too. So one immediate thing that fluoxetine does is this is quite toxic to diatoms. You can clearly see that if you don't add anything, the cells will grow nicely, but if you start increasing the concentration, you actually have a very abrupt shift here. So you have two situations that are barely indistinguishable from the controls, although you can already see that there is a decrease in the growth rate. But if you push to concentrations that are just slightly higher than what has been detected, then you have a dramatic effect. So if this affects growth, definitely it will affect metabolism. And if you look at this, this would be just a comparison, a heat map showing what happens for the sets of concentration that we tested on different biological replicates. And you can clearly see that you have two distinct groups. Each of these small tiles corresponds to one annotated identify the metabolites. So there are a few thousands here. So without entering into detail, you can see that there are very, very dramatic changes in the entire metabolism of this cell. So one of the advantages of using magnetic resonance is because you are ionizing everything at the same time. Of course, you have suppression effects. You don't see everything, but you see a vast diversity of chemical composition that is not affected by the biases that are introduced by chromatographic separation. So you do have a representation of just about everything in the metabolism. Sometimes you have pathways that are better represented than others. That is a work in progress. Maybe one day we will have a single shot where we actually see everything at the same time. So among the things that are affected, of course, is amino acid metabolism. So it's not very much affected by lower concentrations, but it changes a lot, particularly on things that go to tyrosine. So these are not very abundant amino acids, so they are quite critical in metabolic wise, and this is one of that has quite changed. Nucleotide also, terpenols, fatty acids also change a lot. And of course, photosynthesis completely changed, as well as vitamins. But in what concerns photosynthesis, is highly likely to be the causal effect or the consequence of exposure, and then likely explanation for the toxicity that we are seeing here. On the bright side, pigment metabolism is barely affected. So this means that our algae, they will look healthy, but they are not healthy, definitely. So if you were using, for example, optical sensors or satellite images, and this is something that people use to monitor how healthy our ecosystems are, cultures, and so on, these systems, of course, will produce flawed information because what they will see microscopically does not reflect what's happening at the cellular level. So definitely, there are things in the ocean that should not be there, and these things in the ocean go into the food chain, and they have quite a dramatic effect on the entire biosphere. Future projects, of course, we will keep this cooperation with Antarctica, but now we are looking into a different thing. Now we are looking on more long-term effects, not just going there, pick a sample and see what happens. So this is a project that we are developing with the Instituto Dom Luís. So this is the Geophysical and Meteorological Institute. It was founded in the mid of the 19th century. So it has a very long track record of a lot of physical parameters like water temperature, water level, seismic activity, and so on. And basically what we do is that we take a hike on their ocean bottom seismograph. So this is OBS. You can see one here. So basically this is being dropped on the Atlantic Ocean. And our idea with this project that we have some samples, but we haven't started any real analysis yet. But the idea with this hitchhike is because this institute, they drop these OBSs with, I would say, a very high frequency and with a very good mapping of the bottom of the Atlantic. So if you look at the borders of Portugal here, you can see, so this is the platform, 200, 250 meters. And these are some of the locations where these OBSs have been dropped. So in some cases, for example, in abyssal plains, about 6,000 meters deep and something a little bit lower. Out of curiosity, this here is known as the Canyon of Nazareth. So this is almost 7,000 kilometers deep. And this is responsible for the high waves that you see for surfing, because as you can see, it's a very, very deep canyon that goes very close to shore. So the waves go unnoticed, and when they're close to shore, they will raise. This GB is called the Gorange, and this is very close to the surface, 15 to 20 meters at the highest point. So it's a problem for navigation. But this is the region where it's believed that most of the high-intensity earthquakes that wiped Lisbon at least 2 times and a few other cities came from here. So they are very, very close. So this kind of OBSs that you can see here, so they contain inside a lot of sensors for temperature, salinity, very, very sensitive seismographs. These seismographs are sensitive to the touch of a fish. So these guys can actually calculate the mass of the fish that hit the seismograph. And in some cases, there are small fishes, and in other cases, very, very big fish, including at huge depths. And if you look closely here, you will see there are two tubes. So what do we do with these tubes? In some cases, we put tubes that contain growth media, selective growth media. So our idea is that when the OBS comes to the surface, it will bring creatures that live on the depths. And the other thing that we are collecting, of course, is water and sediments that drop from the surface, from the column of water. So this will be the so-called dissolved organic matter just for the water, and natural organic matter, everything that is dropping from the surface. So what are we going to find there? A lot of organic matter, a lot of fingerprints from living organisms for sure, but definitely this is a way to monitor what's happening at an open ocean and not just coastal areas. It's still a starting project, an ongoing project. I hope next time we talk about FHERITALE or another project, that there will be some interesting results to show about this. Of course, I have to thank a lot of people and a lot of organizations for making this work possible. I also thank FHERITALE and David for the invitation to present this work, and I thank you all for your attention. Thank you.
Thank you very much, Carlos, for that very interesting talk. As before, anyone have any questions, please do type them in the Q&A. If not, I can maybe start. I'm going back on my chemistry a little bit, Carlos, so forgive me if I'm ignorant. But thalidomide, we're interested to see that being detected, and obviously that is it's a chiral molecule. It's typically in a racemic mixture, I believe. Can you detect both forms using this mass spec technique? Or is it just...
What do you mean, both forms?
So is again, forgive me if I'm wrong. Thalidomide is a chiral molecule, so the R and the S form.
Yes, chirality is a problem for mass spectrometry. The bottom line is, when we do this ranking of molecules, we work on hypotheses. Let me give you an hypothesis. You are alive. If I get amino acids from you, this will be all in the L form. The moment you die, they will start to racemize. And then I'm starting to detect also the D form. So because these analyses are mainly made with things that are alive, I assume, so there are a lot of assumptions on this. So I do not have, I would say, the highest level of identification. What I have is the formula. What that formula can be, that is another story. So chiral components, can it be distinguished? There are only three possibilities, but the two best possibilities are, first, you use chromatography, a chiral column. The other one, and this is something for which I think there are only 2 or 3 papers in the world, maybe less than that, maybe just 1 or 2, is to use chiral gases and use ion mobility to actually separate chiral species in the gas phase. So this is technically possible. But again, neither of this is compatible with this kind of analysis, because we are aiming at high throughput. What we do is that if we find something that is really, really, really interesting, of course, then we focus on that, and then it's unavoidable that we have to do MSMS to confirm what it is. MSMS is quite tricky because first it depends a lot on the instrument that you are using. It will produce a different pattern. What collision gas are you using? What is the collision energy? What is the geometry of the source? And then most instruments will do just fragmentation by collision. But in magnetic resonance, of course, you have a lot of other possibilities. You can use lasers, you can use electrons. So what is the MSMS that you have? The other reason why we try to avoid it outright is mainly because a lot of MSMS that you actually see are artifacts. So already in this spectra that I've showed, there's a lot of the so-called in-source decay. This is unavoidable. Maybe we can someday relate this in-source decay with the parent ion. So this is a work in progress. There is also one thing that can be done on this type of instruments, but again, we lose the advantage of speed and high throughput. It's called two-dimensional MS. So in this case, we fragment everything that we have trapped inside our cell, these tens of thousands of compounds. Everything is fragmented on the same time. The time to do that, and then we correlate what are the fragments with the ions that actually produce them. But these experiments are quite long. They usually take tens of hours, and then you need also a lot of work to process the spectrum. So it's not something that we do routinely, and I think no one actually does this routinely in the world. But
Sure.
Are we sure? We are not sure. We have a very good hypothesis, and this points us to a direction. Then we go to other techniques to identify.
Yeah.
I think in the future, the difficulty to couple this with LC-MS is that LC-MS, you don't have the time scale when you do direct infusion. You're seeing everything at the same time. So it's difficult to correlate one with the other. You ask me, why, Carlos, don't you use an HPLC? Oh, I have quite a few sitting in the lab that I only use on certain special occasions. It would be possible to actually use the exact same HPLC on two very similar platforms. So I use the Bruker platform. So the natural coupling would be to match this, say, with the Bruker cutoff, and then at the same time, or different times, but split the sample in two, and have on one side LC-MS, and on the other side extreme accuracy from LC-MS. This has been done once for proteomics. So that was called the proteomics cluster. So this is an idea that we can implement here to improve the analytical flow, yes.
Yes, yeah. Okay, I can see another comment in the chat from Ryan. I can read it out. So firstly, very interesting talk and nice photos. And then Ryan goes on to say, you showed the phytoplankton pigment metabolism was not affected by exposure to fluoxetine, although other metabolism was. So if you just look at phytoplankton pigment, then the effect of pollutant exposure may be hidden. Do you think we need to develop new techniques to check on the health of our environments?
Well, large-scale monitoring of the environment is mainly based on some kind of optical sensors. And definitely it depends on what kind of, I would say, multi-wavelength sensors are definitely needed for us to see something. There is maybe going to the non-visible world. I'm thinking about, for example, infrared and other spectral domains that are more related to chemical composition.
Yeah.
I know this was done with yeast cells, and the IR spectra actually is a good telltale of some metabolic activity. So definitely we need better techniques. Because if you are looking just at visible near infrared or near UV, I think you miss or you can miss a lot. And this we are talking about phytoplankton. A lot of things do not have color.
Yeah. Okay.
So,
and maybe one final question, because I'm aware we're slightly over time, but it's a question from me, so I'm going to allow it to be asked. So you mentioned metabolomics and being able to determine what organism was present based on breakdown products. And I wondered how that might link maybe to these artificial substances, and whether we can detect from biotransformation products what organisms are breaking down or acting on organic pollutants, for example.
Yeah, I think that would be quite interesting to do, actually, how these microbes react and how the presence of the pollutants affect the fingerprint, the chemical fingerprint that we see. Of course, we are making the first steps. We are working with individual cultures and also with mixtures. That means consortia that can have 4 or 5 different bacteria.
Yes.
So this is not exactly the real situation that we are going to see in the bottom of the ocean. I don't expect to find exclusive compounds that relate one specific marker for one specific organism.
Sure.
In any case, we will definitely need, and we do this, to use the molecular biology techniques to complement and to give us some certainty, especially at the beginning.
Mm-hmm.
Of course, we use a lot of machine learning too, and we try to use learning techniques that allow us to pinpoint or produce a probability ranking of what current families are there.
Mm-hmm.
But with these things, with the OBS, I'm pretty sure that we are going to find quite a lot of things that hopefully are unknown, and out of these unknowns, hopefully some will be useful, because these things, of course, because they're exposed to toxic substances, they endure selective pressure to evolve. So there could be some selection. The OBS stay on the depths for weeks to months, usually months. So these campaigns can last 3 to 6 months, sometimes more. So they run on batteries, then they come to the surface. Through an acoustic signal, they disconnect from the weight they carry, and they simply float and are caught. So definitely what we will find there, some of these organisms are interesting for this kind of things. Because bioremediation is probably the best option we have. Because through chemical processes, these pollutants are really forever things. So they're going to stay there forever.
Okay, thank you very much. If there's no other questions that I can see, I will call us to a finish and say thank you very much again to both Didier and Carlos for giving us their time this morning, and for two very interesting and engaging talks. As I said, the webinar recording will be made available shortly, and be made available through the FHERITALE website and YouTube channel. And I finally thank you all for joining as well, and for engaging with us today, and look forward to engaging with you in the future as well. Thank you very much.
Thank you very much.
Thank you so much, David. And thanks all our assistants for coming.
Bye-bye.
Bye-bye.